Good morning, everyone. Thank you for joining us for another lunch break webinar session. Today we have a guest speaker with us, which is great news. So I'm just going to do a very brief introduction and remind you where you can find these sessions. And then I'll invite our guest speaker to go ahead and share their screen, and we'll switch to another presentation and have him lead us through it. So just a bit of background, if you haven't joined us before, or even if you need a refresher, the lunch break sessions occur monthly. They're usually on the last Tuesday of every month, and they always happen over the noon hour. To join the mailing list to get the regular meeting invitations, you can contact Brittany at outreach@ctcns.com, and it's displayed there on your screen to join the mailing list. The sessions are always available through online video conference, and that is through the Zoom application. So you have the option of joining from your web browser or downloading the application to your device and joining through the meeting ID. And also, if you're not able to join us over the noon hour, the sessions are always recorded and they will be available to you at any time directly from the Christmas Tree Council of Nova Scotia's YouTube channel. The link is shown there, but you can also search that Christmas Tree Council of NS on YouTube and you'll be able to get to the sessions directly. So it's 12:05 now, and I see we have a few attendees, so we'll get started shortly. One thing I wanted to remind everyone of, because we are in a webinar session, we aren't able to hear participants if they are trying to ask questions. So we ask that you use the Q&A box, which should be displayed at either the top or the bottom of your screen if you hover over it. And you can type directly into the Q&A box, and I'll be monitoring it throughout the session. So if there are questions, I'll take a break and ask our speaker to try to answer some of them for us. So today's speaker is Greg Adams. He has a very diverse history in genetics with forestry production, particularly through Irving, and I'll let him do a more detailed introduction. I think most people are familiar with Greg's work, but I'm going to go ahead and stop sharing my screen so that Greg can come in and take over and guide us through this discussion today.
Well, hopefully the presentation is up and running for folks. Thank you, Jay, for the introduction. And just by way of introduction, as Jay mentioned, I've been involved in genetic tree improvement programs for quite a long time, and not so much with Christmas trees, but over the last year, I've gotten somewhat familiar with that. And so today, I'll share some I guess, an overview of how genetic tree improvement programs work, and specifically relate that to the production of improved balsam fir Christmas trees. So in terms of an outline, I'll talk about the objectives of tree improvement programs, genetic variation in balsam fir, traits of interest for Christmas tree producers, how genetic gain for individual traits works, and the relationship between different traits, genetic variation related to production strategies, the status of tree improvement in Christmas trees in the Maritimes, and some value-added suggestions with some just some summary notes at the end. So objectives of tree improvement. Planting trees is an expensive endeavor, as anybody would know, with preparing the site, purchasing the trees, planting the trees, and then looking after them. And tree improvement programs seek to improve the return on this tree planting investment in a broad long-term sense. In Christmas trees, there are obvious aspects like increasing the grade and value of trees and perhaps shortening the time to market. There's other less tangible benefits, things like improving resilience to insects or disease attack and environmental stresses, or reducing cost of production. So, possibly things like less shearing and these sorts of things. Compared to tree improvement for reforestation, which I've spent the last numbers of decades doing, Christmas tree improvement has a quick return on investment. When you look at a somewhere between 6 and 10 year rotation on a Christmas tree lot, I'd have to say that I'm somewhat envious of that, having worked with trees for a long time with a 35 to 45 year return on investment. So reviewing genetic variation for Christmas trees, and I'll speak specifically about balsam fir. You know, we're not going to have time to get into other species, but with respect to balsam fir, it occurs at different levels. One is regional variation, and it doesn't show up great on this slide, but this is a range map for balsam fir, and it shows that it goes from Saskatchewan through Manitoba and Ontario, Quebec, and then throughout the Maritimes and Newfoundland and Labrador, and down into the States. It goes into Minnesota, Wisconsin, and then you know, upstate New York, and Maine, New Hampshire, Vermont. So these places, that's the natural range. Genetic variation at this level tends to be most associated with climate adaptation, and it occurs over the climate gradient that you would expect. That's a big piece of geography in a lot of different climatic regions. The second level of variation is stand level. So that's the variation associated with a specific stand of balsam fir trees. And there's quite often, there's some level of stand level variation. Some of it's related to stand history, but it's one component of the variation. And lastly, the within stand level. So that's the variation in individual balsam fir within a stand of balsam fir trees. And this variation is always large. And so when you, and really it's the improvement of balsam fir Christmas trees involves harnessing both that stand and within stand variation within our region. So traits of interest for Christmas tree producers, and again, in spending some time with the Christmas Tree Council in the last year, as near as I can understand, you know, the traits are things like crown form, growth rate to a certain extent, pest resistance, needle retention or needle abscission, and then a couple of environmental type or adaptive type traits like late frost tolerance and early winter hardiness. And I'll speak a little bit about late frost tolerance. I know people had a very, very bad experience a couple of years ago with a late frost in the middle of June. And yes, wouldn't it be nice if you could do something through tree breeding to mitigate that type of damage? So frost tolerance, late frost tolerance is related to flushing. And certainly flushing dates in balsam fir, it would be heritable. The only thing is the range and variation is not very great. So I think you know, some level of late frost tolerance could be could possibly be developed, but you know, you're probably better off in looking at avoiding frost pockets and things like that when you're planting trees. And because you know, the event that you had several years ago was pretty serious, and where it's tied to flushing, pretty much everything will have flushed well before then. There's another element of adaptation that may be of value to you, and that's early winter hardiness. So I think there's probably more variation to the development of winter hardiness in balsam fir. And it's well understood that needle retention is quite associated with that. So that's another possible trait that could be of value to producers. So genetic gain for individual traits. To make genetic gain in planted seedlings, you need to meet some prerequisites. The first is there has to be variation in the trait that you want to improve. I think that's pretty obvious. If there isn't any variation, you're pushing a rope trying to improve it. There needs to be a genetic basis for the variation. So, and in other words, the trait has to be heritable. There's some things or some variation that you'll see that's just a matter of environment. So, but to make genetic gain, it has to be heritable. The last thing is you have to have a large enough population to select from. So the way you calculate gain is the amount of variation times the heritability times the selection intensity. So are you picking the best 1 out of 5, or are you picking the best 1 out of 1,000? Clearly, you're going to make more gain if you're picking the best 1 out of 1,000. And I'll skip over this, but you know, the efforts in tree improvement needs to be sized with the size of the planting program that you have. So relationship between traits. The more traits in an improvement program, the more complicated it becomes. Some traits are genetically correlated. So for instance, you might expect the taller the tree, the bigger the diameter it will have. This relationship between traits is important for a couple of reasons. Firstly, if 2 traits are negatively correlated, then selection for either will result in negative outcomes for the other trait. So that's a highly undesirable situation. And hopefully the traits that you're interested in, you're not going to experience that. If 2 traits are positively correlated, then selection for one trait will enhance the other. So that's great. You don't even have to bother with 2 traits. You just pick one and select for that, and you'll improve the other. The other more common situation is if 2 traits are not correlated, then gain is possible for both, but you have to have more to pick from to get the same potential gain for each. And there's a good example of that came from the Smart Christmas Tree Project. And this is looking at 2 different traits, and the dots represent individual parents. And the 2 traits is needle abscission and aphid rank, which is susceptibility to balsam woolly adelgid. So if you look at the areas that I've got circled, you've got a number of parents that exhibit. Clearly, if you look at the whole scatter plot, there doesn't seem to be any relationship between needle abscission and aphid susceptibility. But if you look at the circled areas, there's a group of parents that have high retention and high susceptibility. There's another group that has high retention and low susceptibility. So if you were looking at just strictly selecting the best 3 of those parents for needle retention, one of them would be quite resistant to adelgid, and the other 2 would be susceptible. So that's not very good. Similarly, if you were just interested in aphid rank, you know, the top 3 parents, all 3 of them are lower than average for needle abscission. So you want this group with the high retention and low susceptibility. But if you want to get the same amount of gain for each of the traits, it means you just simply have to have a lot more to pick from to get the same number of parents with the better combination of traits. So genetic variance related to the production strategy that you choose. So when you look at a tree in your Christmas tree lot, what you're looking at is what's called a phenotype. So you're looking at a tree in the woods, and that tree that you're looking at is a combination of genetics plus environmental factors, the whole history that tree's experienced through time, and you know, all site factors that are associated. And normally in tree improvement, you do some form of genetic field testing to measure the genetic variation in a population for the traits of interest. So what you're trying to do is you're trying to tease apart those 2 components of genetics plus environment. Most traits are controlled either positively or negatively by multiple genes, which each contribute to the genetic value of the individual. Genetic improvement programs involving seed production, like seed orchards, rely on the sum of positive genes contributed by parents to the population, and this is termed additive genetic variance. So the seed orchard programs all around the world, they're trying to lever additive genetic variance. Another form of genetic variance results from the very specific combination of genes in an individual, and this is called non-additive genetic variance. Every time you go through sexual reproduction, producing seed, the genes are recombined. So you can't, because the non-additive variance is associated with specific combinations, you can't utilize non-additive genetic variance in seed orchards. It's only possible through cloning. The next thing I'm going to I'm going to go through sort of the traditional tree improvement approaches and give you a sense of where things are at with genetic improvement in Christmas trees in this area. So the traditional tree improvement steps starts with selecting the superior phenotypes for important traits. So you go around across the region and identify trees and growing in Christmas tree lots that you think are pretty good for important traits, and then taking those and grafting them to establish seed orchards. So by taking those individual trees, taking cuttings from them and grafting them onto rootstock, you're basically cloning those individuals, and then you clone, you make a number of grafts from each of the trees you've selected and establish seed orchards with those cloned phenotypes all mixed up. The next step is to conduct breeding and field testing or clonal assessment to understand genetic variance and to identify genetically superior trees in the forest. So we're going to talk quite a bit about that. Then the next step is, once you, through whatever means of breeding or clonal assessment in the seed orchard, identifying the less superior parents and genetically rogue them to improve the genetic quality of the seed in the seed orchard. So then the last thing in most traditional tree improvement programs is, if breeding and progeny testing is being conducted, you would then select the best individuals from the best families and initiate the next breeding cycle. And if you look all around the world, you know, some of the oldest tree improvement programs are into the fourth generation. It takes a long time to turn over a generation in traditional tree improvement programs because of the long testing required. But you know, with each generation, you make more and more gain. If you turn around and compare that to corn, they turn over a generation a year, and corn's been bred through thousands of cycles of improvement. If you look at where things are at today, so you can see I put check marks beside select superior phenotypes and graft and establish the seed orchards. So those have been done. There's been some work done through a couple of different efforts, including the Smart Christmas Tree Project, to evaluate parents in the seed orchard. But there's been very little, there's been some, but not very much breeding and field testing underway to identify which parents are superior in the orchard. There hasn't been any genetic roguing in seed orchards to improve genetic quality. And of course, there's been no work on the next cycle of breeding. So going through those individual steps, there was very good, strong selection for the best phenotypes in Nova Scotia, but also in New Brunswick as well. And there were somewhere around 400 selections initially made. And since then, I believe there's been other selection efforts that have been underway in Nova Scotia as well. The early efforts were made in collaboration with producers and provincial government and tree improvement staff. And you know, it started with identifying the best trees in the field, and usually that's in the form of some kind of checklot or a checklist where you would go through the traits and evaluate potential candidates, and based on the evaluations, determine which trees you believe make the grade as a Christmas tree selection. So this is the writing you see on the side is an actual evaluation of a tree in the woods, and it's covering things like pests and deformities, growth rate, foliage condition and needle retention, form and branching, and going through the process to determine a score. So the next step, the establishment of seed orchards. Grafting was conducted and orchards were established in the 90s, and the orchards at DeBert and Kingsclear in New Brunswick have been maintained and are an excellent asset, and no roguing is being conducted. From a grafting standpoint, balsam fir is great. It's one of the easiest trees around to graft. So genetic evaluation of parents. So to date, there's been minimal breeding and field testing. But in general, the way this works is when testing is done, they're replicated across several sites and statistically designed to evaluate the genetic worth. And what's usually done is they're planted over some sort of site gradient and weather gradient. So you don't, you always want to make sure that what you're selecting for one area is suited for another area. So that's the purpose of this replication across sites. For some traits, though, multiple grafts of each parent could be evaluated in the orchard to provide an estimate of genetic worth for some specific traits. So work has already been conducted in the SMART Christmas tree program on needle retention and some on pest resistance. And this can certainly be used in future selection and testing. I got a couple other examples of potential evaluation in seed orchards that are possible. So grafted trees in orchards are not suitable for evaluating traits like growth because they're grown very open and in very open conditions for seed production, and they are grafted trees, so you can't necessarily count on growth traits by evaluating seed orchards, but can be useful for things like pest resistance, adaptive traits, needle retention, and maybe even crown form. And I took a couple of pictures in the Kings Clear orchard in Fredericton a couple weeks ago, and that illustrates this. So if you look at the photo on the left, you've got a tree in the seed orchard that's clearly been heavily impacted by balsam woolly adelgid, and the one right beside it, not so much. Similarly, on crown form, I'm not a Christmas tree producer, so the folks listening are in better shape to evaluate, but in my eye, the one on the left looks like it has superior crown form for a Christmas tree compared to the one on the right. In order to do this, it's not just a matter of finding trees with the right trait. You really need to systematically evaluate all the ramettes in the orchard, score them, and then analyze the data. I will make a note here that sometimes crown management is done in a seed orchard, and that could interfere with this potential. So quite often in seed orchards, the trees are topped so that it makes it easier to pick the cones. I have another example. This is the balsam fir Christmas tree orchard at DeBert. And as you can see, the trees are quite big. It's difficult to collect the cones. And in 2019, some helicopter cone collection effort was done by lowering a basket over the top of the crowns of trees and raking off branches. So that clearly interferes with the appearance of the tree and possibly the evaluation of the parents. I'm going to mention other methods of genetic improvement in Christmas trees besides orchards, because I know there has been a lot of work done in Nova Scotia on this technology through the Smart Christmas Tree Project. And the method that most effort has gone into is the use of somatic embryogenesis. And so I just wanted to provide a couple of slides showing how that fits in, but we're not going to go into any detail. Basically, somatic embryogenesis allows for capturing genetic gain from non-additive as well as additive genetic variants. And the way it starts is you identify the trees that you believe are the best in your seed orchard. You make control crosses, so crosses where you know the mother and the father for all the seedlings, and make a number of crosses amongst a number of parents. And out of that, you get a small handful of seed from each one of these crosses. And from every one of the seeds, you take out the embryo and put it on a tissue culture medium where in the right conditions, it will start to form cell masses that are kind of like stem cells that we hear about all the time in human disease treatments. So you take these cells, all the cells in here have the potential of developing into a whole tree. And this material can be frozen in cryo storage for basically as long as you want to store it. At the same time, you take some of that callus from every single one of the embryos that you've excised, put it on a different medium that allows embryos to mature. And these are mature embryos that all come from somatic embryogenesis callus, and they're genetically identical. You can take some of those trees and produce seedlings from them, establish your tests in the field, and then based on the results of those tests, you can identify the very best combinations, so both the non-additive and additive genetic variants, go back to the cryostorage, pull them out, and start mass producing them. So that's the technology that folks have been working on. So the advantages: when you make a cross between good parents, the progeny will be above average overall, but there's large variability within the families. I've often told people that nobody's ever come up to me on the street and said, "You must be Owen Adams's brother. I can tell by looking at you." Well, you know, we don't look the same. So there's huge variation within families, even with the same mother and father. You can preserve the individual clones by cryostorage while field testing is conducted. And then once proven, those clones can be brought back for mass production. With enough clones, there's a higher probability of finding clones of high value for all the traits of interest. So just like we saw on the graph, you're going to find some clones that have good growth rate, good needle retention, good pest resistance. That's what you're really after. But it's all a numbers game. The more you have to pick from, the more gain you make. So the disadvantages: not every seed from every family responds, and it requires skill and effort to produce the many lines required and enough clones to pick from. It requires more effort to grow a somatic seedling, going through all those steps compared to just collecting cones and sowing seeds. Due to these higher SE production costs, you must ensure that you have sufficient gain to make it all worthwhile. So that's the two approaches that we'll talk about, that we've talked about, but we're going to deal mostly on the seed orchard options from now on. So looking at the state of where tree improvement's at, I make a couple of suggestions on improving the value of Christmas trees based on that current status. The first note I make is about data management. To be efficient and scientific about this effort, you need to compile a single database for all the selections that are available with any of the known genetic attributes that we have. And to do this properly, somebody needs to own it. So you know, somebody needs to look after that database and be capable of managing that and doing analysis. I think an immediate step would be to look at all the ramettes of all parents in orchards for insect damage and crown form. There are a couple of simple things that could be done to increase your gain. It might be also worthwhile to look at fall hardiness from a needle retention standpoint, and maybe you want to consider additional needle retention studies. Would all provide useful data to improve the value going forward. I think it would be a worthwhile effort to bring Nova Scotia and New Brunswick selections together for testing. And you know, that starts with getting the provincial tree improvement folks on board and with a clear plan of how it could be approached. And the quickest way forward, I believe, would be to collect open pollinated seeds. So just collect cones from individual parents. And certainly you could focus on parents that have already been identified to have good needle retention, and you know, possibly further evaluation of early fall hardiness and good crown and insect tolerance, and take that information and do the analysis and decision making around trait trade-offs. But the whole idea would be get a bunch of open pollinated families and get them out into tests. Again, there's a fair bit of tree improvement genetics expertise in the region, and it's always a good idea to seek some collaboration in some of those areas. You know, in Nova Scotia, you might want to consider establishing a couple of test sites, one on the South Shore and one between Truro and Antigonish, where a lot of the Christmas trees are produced. And when you look at establishing a test site, you're looking for a uniform site, because what you're trying to do is isolate the genetic effects. So it's not that the trees have to have a uniform site to grow on, but what you want to do is eliminate as much as the environmental effects as you can to get a good signal on the genetics. You'd usually do a replicated design, something like 20 replicates with one tree from each parent in each rep. That's the sort of design that would typically be used in this kind of testing program. Now on the production side, seed from established orchards should be used as much as possible. And something you can do to improve cone production is there's flower induction treatments with gibberellic acid that help the trees flower more. Certainly focus collections on parents that have already been determined to have good needle retention and pest tolerance. This will give you some immediate genetic gain for these traits based on the additive genetic variants. The trees in Debert at the old seed orchard there, they're getting large, making cone collection a lot more difficult. And you know, it could be a useful thing to consider regrafting that. Some of this has already been done at Plumdale. I'm not sure the exact extent, but you want to make enough grafts so that you can generate significant amounts of seed over time. So that's something that could be considered. And if there's no further use in a seed orchard, or if an orchard is going to continue to be used, for instance, you know, the King's Clear orchards are somewhat they're a little bit younger. They aren't quite as I guess as they're plenty tall, but it's a place where you could consider genetically roguing those based on the assessment of the grafts, and then that would increase the genetic gain from those orchards as well. Again, that would have to be in collaboration with the provincial government who operates the orchard. So just in summary, you know, there's a strong case to build on. There's been solid efforts made in Nova Scotia and New Brunswick in selection, grafting, and orchard establishment, and it's provided a foundation for improved Christmas trees today and further improvement into the future. Requires long-term commitment. The organization, the Christmas Tree Council, needs to develop continuity and expertise, and it would be a good time for the Christmas tree producer groups to develop these collaborations with provincial tree improvement programs. And certainly expenditures on tree improvement need to be commensurate with the size of the industry, and specifically the number of trees that are planted every year. So if not, you know, if the numbers are relatively small, then it's more of a basic program, and the larger the number, then you can contemplate additional approaches that increase the cost but also increase the gain. So with that, that's the end of my slides, and I'm certainly open to answer any questions that have come in. Thanks.
Okay, thank you very much for that, Greg. I think we all appreciated it. So far, I don't have any questions popping in, which is usually a sign that your presentation was well understood and well accepted. So I'll ask you to stay on for a couple of minutes just in case people are having trouble typing into the question box. But if I don't get anything in a few minutes, you'll be free to go.
No problem.